Hello, and welcome to part 3 of chapter 4, bacterial growth or microbial growth. In this section, we're going to look at some of the different nutritional factors that are required for growth. In part 2, we looked at environmental factors. So now we're going to move into nutritional, or sometimes referred to as chemical factors. First thing I want to talk about are some of the required elements for bacterial growth, microbial growth, and they're pretty much the same that we see in eukaryotic organisms. Of course, we have the primary big four, the carbon, oxygen, hydrogen, and nitrogen. Then we also have, for some amino acids, such as cysteine, we have sulfur, of course, phosphorus, and potassium, all of these vitamins or minerals. And then we move down into the very low concentration compounds, such as the trace elements. And when we think about nutritional factors, one of the things we have to take into consideration is whether our organisms are heterotrophic or autotrophic. And so trophic levels are based on the source of carbon that organisms use for energy and for building blocks. So heterotrophic organisms use organic carbon in its already existing organic carbon state, such as a carbohydrate or an amino acid or a lipid and such. Autotrophic organisms are capable of taking inorganic carbon and using those inorganic carbon sources, such as CO2, probably the most common inorganic carbon molecule, is taken in by organisms and then used to build the precursor building blocks to organic molecules. So heterotrophic organisms recycle organic molecules, whereas autotrophic organisms take in inorganic and convert them into organic molecules. And so one of the things that are important, one of the functions or one of the properties of some bacteria that are important is their role in the carbon cycle on Earth, in that recycling of carbon from one form to another. Another important role that we need to talk about for bacteria, prokaryotic organisms in environmental microbiology is their role in the nitrogen cycle. So in the nitrogen cycle, bacteria are critical, and we would have no nitrogen cycle without them. And the nitrogen cycle really includes two primary phases. Nitrogen fixation is one, and then denitrification is the other. So in nitrogen fixation, bacteria are capable of converting gaseous atmospheric nitrogen, N2, that's what we're looking at right here. They can convert this into a usable form of nitrogen, ammonia in particular. This usable form of ammonia that is now available to plants for growth. Remember, plants are eukaryotic organisms and they're going to need a lot of nitrogen as well. So in nitrification, we can convert atmospheric nitrogen into usable forms of nitrogen. There are a couple of different usable forms. The first is, of course, ammonia. So you can see here in our little drawing here of a nitrogen cycle, we have nitrogen-fixing prokaryotes that can convert that atmospheric nitrogen into ammonia. That's going to be observed by the plant roots. We have certain classes of plants that called legumes that have nodules on their roots, and those nodules are basically housing communities for prokaryotic organisms that are nitrogen-fixing. So those organisms are going to absorb that NH3 that's been converted and is found in the soil, and they're going to convert it into NO2 and NO3. So these are our nitrites and nitrates are also considered usable forms of nitrogen for other non-legume plants. Now then we move into the denitrification stage, and in denitrification, the nitrates get converted back into atmospheric nitrogen. And then that atmospheric nitrogen will be recycled by our nitrogen fixing prokaryotes. So you can see this is this kind of nitrogen cyclic process where we're providing, or not us, but prokaryotic organisms are providing nitrogen in multiple forms. By rearranging it, they are providing nitrogen in multiple forms for different classes of plants to grow, other organisms that may need nitrogen for growth, and then of course putting nitrogen back into the atmosphere for that recycling process. Now, when we think about growing bacteria in the laboratory, we have to think about multiple growth factors. And we oftentimes will kind of drop bacteria into one of two classifications. They are either what we call fastidious or non-fastidious organisms. So a fastidious organism has a very complex nutrient requirement. They have a whole lot of different nutrients they need in different concentrations in order to grow. So I use the analogy of a buffet. A fastidious organism is going to want to eat, going to want to go to dinner at a buffet because they need to have a multitude of different things available to them to choose from. A non-fastidious organism is one that is very specific about its nutrient requirements, and they oftentimes have the ability to biosynthesize their own complex molecules from very simple biomolecules. So in this case, a non-fastidious organism may be one that wants to sit down with a menu and order very specific simple meals off of a menu in order to create one really large complex meal. In this case, something like converting glucose into other raw materials available to cells. So we have fastidious organisms. They have very complex nutrient requirements. E. coli is an example of a fastidious organism. And then we have non-fastidious organisms that have much simpler but very specific growth requirements. And so an example of one of those would be strep pyogenes or strep facialis. Now, in these trophic levels, we talked about heterotrophic and autotrophic in those trophic levels. Well, those get further classified into kind of subclasses in what we call nutritional diversity. So some microorganisms are considered photoautotrophic. Think of cyanobacteria. These are molecules that have these are prokaryotic organisms, not molecules, excuse me. These are prokaryotic organisms that have the ability to synthesize their own biomolecules from CO2 using light as an energy source. So these guys are basically photosynthetic bacteria, and the ones that we would think about in this would be the cyanobacteria. Photoheterotrophic organisms will use light energy to take simple carbon molecules or simple organic molecules and use light energy to put those organic molecules together to build more complex organic molecules. A chemolithoautotroph is one that uses inorganic carbon for energy and derives its carbon directly from CO2. This is known as a chemoautotroph. And then the chemoheterotrophs, or chemoorganoheterotrophs, we call them chemoheterotrophs, they use carbon sources in their existing state as their primary source for biosynthesis. They are recyclers. We, as warm-blooded mammals, are chemoheterotrophs. Most eukaryotic organisms will fall, and when we talk about animals, will fall in this chemoheterotroph trophic level. So when we look at bacteria, look at growing bacteria in the laboratory, we have to take a lot of different things into consideration. But we can determine many of these considerations based on where an organism is coming from in nature. So there's a large diversity of nutrient requirements, and because of that, we have this really wide range of different kinds of media. Now remember from our previous lecture, our two basic forms are solids, which are agars, and then we have broths, which is basically water plus supplements, right? And so both of these will contain lots of different supplements. Now, what combination of supplements are added to the agar or the water depends on where this organism is coming from and what its nutritional diversity may be. So some things that we need to consider in order to promote the growth of microorganisms in the laboratory. That's what cultivation means. Cultivation means that we are purposely growing these microorganisms. So it's important in cultivation of prokaryotes that we address all of the different nutritional, chemical, and environmental requirements for growth. So of course, temperature, what are the nutritional requirements? What about oxygen? That's another environmental requirement. How long is it going to take this organism if I'm not growing it at the right temperature, oxygenic, or nutrient requirements? Am I going to inhibit and slow down or speed up its growth? Does it grow better in a solid or a liquid? If I find this organism in soil, I'm probably going to have better luck growing it on a solid media. But if I found this particular organism is found in aquatic environments like a lake sample or an ocean sample, seawater, then I'm probably going to have better luck growing it in a liquid culture. And then finally, we have stock cultivation. What is the purpose of growing this in the laboratory? Am I using this for research? Is this a control organism, so I need lots of it for every time I run a test? Or is this an organism that I'm testing for certain metabolic characteristics or that I'm trying to identify? All of these different things, all of these different considerations must be included when we're trying to determine the best way to grow this organism in the laboratory. So that concludes this section of Chapter 4 on cultivation of prokaryotes. In the next lecture, we're going to talk about the different classes of media and how those media are used in the identification of bacteria, as well as in diagnostics and in environmental microbiology. See you in the next one.